All right, so thank you, Mehrdad, for organizing this wonderful event. So what I'll be talking about today is work done by Matthew Golub, and he is co-advised by Professor Stephen Chase and me. And all three of us are here at this meeting, and so feel free to flag any of us down afterwards to chat. And also, Matt will be presenting a poster here, so feel free to go there as well. Okay, so to introduce how we conceptualize internal models, let me just give you a simple illustration. So this is something that happened yesterday at lunch. So I ordered some clam chowder, and as I was reaching for it, I realized my neighbor ordered lobster, and I really wanted that, and so I reached for it. How did I successfully change my mind and get the lobster without misaiming? Okay, so if you think about what's going on, when my hand is at this point, because of sensory feedback delay, my brain only knows where my hand was a bit of time in the past, right? And so if I were to aim my hand from this old sensory feedback directly to the lobster, then I would miss the lobster, right? However, if instead my brain used a more intelligent strategy, right, whereby I take my outdated sensory feedback and then update it using motor commands that my brain knows that it's sent since then, right, using a so-called forward model, then I can come up with an estimate of where my hand currently is and then aim straight upwards, right, and successfully reach for the lobster. All right, so many people have studied internal models in the field, and many of them are here in the room today, right? And many of these studies have been on the level of behavior, right, and on the level of single neurons with behavior. So the motivation for today's talk is the fact that we know that thought processes are multidimensional, right? And presumably, the internal models also operate on these multidimensional thought processes. But if we look at behavior only, right, then that's sort of a low-dimensional readout of what our brains are doing, right? And then a similar story is with single neurons, right? And so to try to get at a richer representation of internal models, what we set out to do was to look at neural population activity and to ask whether we could extract a representation of an internal model that could operate on the time scale of tens of milliseconds and also on the spatial scale of individual neurons, and many of them. Okay. So the outline here is there's two parts. So first, I'll talk about using brain-computer interfaces as a tool for studying internal models. And then next, I'll talk about extracting a rich internal model from neural population activity. Okay. So if we look at the native arm control system, right, there are many thousands of neurons that project down the spinal cord. And the effector dynamics are nonlinear with muscle dynamics and so forth, and high dimensional, right? We are capable of multidimensional movements. And there are many types of sensory feedback, including vision, proprioception, et cetera. Okay, so this makes for a very complicated system to study. And what we're going to propose here is to use brain-computer interfaces as a simplified motor control system that has closed-loop feedback, okay, to study internal models. So a brain-computer interface, right, is one in which we record from many neurons at the same time, for example, using an electrode array. We then pass the neural activity into a so-called BCI mapping, which translates the neural activity into movements of a computer cursor, and the subject gets visual feedback on a moment-by-moment basis about where the cursor is. Okay, so if we look at this closed-loop motor control system, what we see is that it's a simple version of the system we really want to understand. Okay? And by maybe using a BCI system, we might be able to get some foothold on questions like internal models. So if we make this comparison, we typically record from tens of neurons using these multi-electrode arrays. And so in this case, we have the full set of control signals, right, that are controlling the cursor by definition. Right? It's only those neurons from which we're recording directly that can affect the cursor motion. Right? There are many other neurons that are playing a role, right, in this, right? But ultimately, the signals have to pass through the neurons that we're recording for them to affect cursor motion. Okay? And then furthermore, we get to define the relationship between the neural activity and the cursor movements. Right? And so we can define them to be linear, right? So there could be a linear relationship between firing rates and cursor velocity. And in this case, we have low-dimensional movements because it's a cursor moving on a computer screen. Okay? And then furthermore, we can restrict sensory feedback to be vision only, right, in this experiment, which makes things simpler. Okay, so what we're going to set about doing is to use a brain-computer interface to study internal models. So these experiments were done by Steve Chase while he was a postdoc in Andy Schwartz's lab at the University of Pittsburgh. And so just to give you a quick snapshot on the details of the experiment, so we used one of these 96-electrode Utah arrays implanted in the motor cortex of macaque monkeys. There were 54 experimental sessions combined across 2 monkeys. I'll show results individually. And also, we recorded 15 to 40 single and multi-units in each session. And the mapping between the neural activity and the cursor movements was a population vector decoder, okay, with 33 millisecond bins. Okay, so just to give you sort of one more layer of detail on what it is that we're sending into the BCI mapping. Right? What we have is recording from many neurons at the same time. Right? Each of these tick marks is a spike. Right? And we're looking at the activity across all the neurons that are being recorded and taking a spike count, right, for each of the neurons in this 33 millisecond bin. Okay? That's the control signal that the animal is sending at this particular time step. Okay? And then at the next time step, it sends another vector, another control signal, et cetera, et cetera. Okay. So just to give you an idea of the level of control that the monkey is showing, I'm going to play a video. Right? In this video, the green dot is the cursor, which the animal is moving just by modulating its motor cortical activity, and the blue dot is the target. Okay. So what you see is that these movements are relatively straight, and they acquire the targets in less than a second. Okay? And so they're about half as good as real arm movements. Okay. So in this experiment, there were 16 targets around a circle, right? And you see that each of these movements are relatively consistent from trial to trial. All right. So the first big question that we're going to ask, okay, is whether these animals use an internal model during BCI control. Right? So no one has asked this question before, and before we can use a BCI to study internal models, we first need to address this. Okay. So I'm going to show you 2 lines of evidence that suggest that these animals might be using internal models. So the first is by asking the following question. Okay. So let's say that this is the cursor trajectory, that this is what happens on the screen, right? And the target is here. And we're going to ask, at any given moment in time, the direction in which the animal sends the cursor, right, which in this case, right, is in this direction, if we look at the tangent of the cursor trajectory, okay, whether that command is more appropriate at the current time or some time in the past. Right? Because remember, when the cursor is actually here, the animal doesn't know that the cursor is here. Right? Its most recent sensory feedback is sometime back here. Right? So the question is whether it aims from its most recent sensory feedback, which is outdated somewhere here, or whether it does some sort of internal prediction right, to estimate that the cursor is actually here and then aims appropriately. Okay. So what you're going to see in the next plot is on the horizontal axis will be time delay. How far back in time do we shift this arrow? And on the vertical axis will be angular error, right? Which is if this arrow goes right to the target, that would be 0 degrees error. But if the arrow would miss the target, then you would get a positive error. Okay. So if hypothetically, right, if the subject aimed from its most current visual feedback, right, which is old and stale, right, then what we would see is that the angular error would look something like this, which is that the angular error would be smallest at the visual feedback delay, whatever it is, right, which people estimate to be around 100 milliseconds for motor cortex. Okay. What we saw, okay, in our analyses with real data was that this minimum was actually at 0 milliseconds, right, which indicates that the command that the animal is sending at any given time is most appropriate for the current cursor position, okay, that the animal doesn't actually see, right, rather than its most recent sensory feedback. Okay? And so this suggests that the subject uses an internal model to predict the current cursor position. All right, so that's the first line of evidence. The second line of evidence is by leveraging an aspect of BCIs, which is changing the BCI mapping, right? Because we as experimenters, we can change the relationship between the neural activity and the cursor velocity. So that's what we did. Okay? And what I'm showing you here is the first trial after this mapping was switched. Okay? So our hypothesis was that the animal on this trial would still be using, right, an internal model appropriate for the first mapping, right? Because the change was made unbeknownst to the animal. Okay, so in black here is the cursor trajectory that the animal saw on the screen on this first trial. Okay, and what we did in post hoc analyses was to pass the same neural activity through the first mapping, right, to see how appropriate it would have been, right, for the first mapping. Okay, so let's say we take, we start at this time point, right, on the actual trajectory, okay, and if we pass that, the neural activity in the time steps coming up through the second mapping, we of course trace out this trajectory. However, if we pass the same neural activity through the first mapping, we get this different trajectory, which we call a whisker. And what we notice here is that this whisker points right to the target, right? As if the neural activity that the animal was sending, right, was appropriate for the first mapping, but not for the second. Okay, we can do this, okay, at all the time points. Okay, and the thing that jumps out at us is that even though the cursor trajectory at times is not going directly to the target, right, and at other times is, right, all these whiskers tend to point right to the target, right? And so this is an indication that the subject continued to use an internal model that was consistent with the first mapping, right, even when the second mapping was the one that was actually under operation. Okay, so with these two lines of evidence, okay, what we set out to do was to try to extract a rich internal model from neural population activity during closed-loop brain-computer interface control. Okay, so I don't have time to get into the details of how we did this, but let me give you the main ideas. Okay, so the BCI mapping that was used took on this form, okay, where x is a state of the cursor, right? So this is position and velocity, okay? And u is this vector of neural activity, the spike counts, right, given off by the neurons that we're recording from simultaneously at one time step. Okay? And then A, B, and little b are the matrices that define the mapping. Okay. So what we attempted to do was to extract an internal model, okay, of this form. Okay? So you notice that takes exactly the same form as the mapping itself. Okay? And we did this both out of mathematical convenience, okay, and due to the fact that if the animal learned a perfect internal model, right, it should fall within this family. Okay? And so the parameters that define the internal model are A tilde, B, big B tilde, and little b tilde. Okay? The X tilde here is the subject's internal conception, okay, of where the cursor is going. Right? Some sort of internal tracking state variable. Okay? And then you notice here that the U doesn't have a tilde on it. Okay? It's the same U as up here. Okay? He's using his neural activity or his motor command that he knows that he sent, okay, to try to do this internal tracking. Okay. So the internal model estimation problem, okay, can be succinctly stated as follows. So what we can observe in the experiment are the cursor trajectories, right? This is what actually happened on the screen, right? The neural population activity that we recorded, right? This is the activity that was controlling the cursor. And then also knowledge of task goals, right? Where was the target? Okay. And what we seek to find, right, are the parameters of the internal model, A tilde, B tilde, and little b tilde. Also, the internal state predictions, right? Every moment in time, right? Where did the animal think the cursor was? Okay, this is the X tilde variables. And also the visual feedback delay, which we call tau. Okay, so you might be asking yourself at this point, right? We're recording using electrode array in motor cortex. How do we even stand a chance of extracting an internal model when we are recording from an area that presumably is not the main area involved in implementing this internal model, right? Probably cerebellum is, right? And furthermore, we have no evidence that we're recording corollary discharge, right? Okay, so the rationale for this is the following, right? That if an internal model is being used somewhere in the motor control system, and that the neurons that we're recording from in the motor cortex are downstream from that internal model, then presumably the activity that we're recording reflects whatever computations are being performed by the internal model. Okay, so by looking at the neural activity, we should be able to deduce something about the internal model. And secondly, we take the M1 activity that we record as a proxy for corollary discharge, right? So if the M1 activity is reflecting the subject's intentions, right, of where to move the cursor, then presumably the corollary discharge is primarily carrying that information as well. Okay. All right. So just to give you some intuition for what this internal model estimation procedure does, okay? So let's take one particular time, okay? So let's say that the cursor, right, is moving on the screen in this direction, okay? And let's say that the animal is sitting at this time, okay? Or the cursor is here, right? But the animal doesn't actually know that the cursor is here, right? The most recent sensory feedback that the animal received was, for say, here, okay? And so what the animal should do is it should take its most recent sensory feedback and then propagate it forward in time using its internal model and the neural activity it knows it has sent since then, okay, to come up with an estimate, an internal estimate of where it thinks the cursor is. Right? So you notice here that there's a mismatch between where the cursor currently actually is versus where the animal thinks it is. Right? And so what the animal should do at this time point, right, in terms of how it decides what is the next neural command to send out, is to then aim straight to the target from this point, right? Not this point. Right? Okay. And so if that same neural activity that the animal sends, right, to move from the current internal estimate towards the target, that same neural activity vector u of t, when passed through the actual mapping, might send the cursor on the screen in this other direction. Okay. So this is the intuition, right, behind this internal model estimation problem. Right? We want to find these parameters of the internal model, this a tilde, big B tilde, and little b tilde, okay, such that at each time point, this is true, right? That using the same set of parameters at any moment, right, the internal estimate of the cursor position is the one that's appropriate for sending the cursor straight to the target. Okay. So we took many trials, right, from one session. We did this across many sessions, but we analyzed one session at a time. We took many trials, right, and we fit this internal model, okay, to the data. Okay? And we did this using an exact EM algorithm. And part of this procedure, right, was to estimate this sensory feedback delay. Right? So in the picture that I showed you, how many time steps back should we go, right, for this estimation of that whisker to come out? Okay? And that's a model selection problem. Okay? And what we found was that a feedback delay of 3 time steps, which corresponds to approximately 100 milliseconds, was most appropriate for the data. Okay? And this is consistent with previous studies that show that motor cortex reflects visual input after about 100 milliseconds. Okay, and then all the results that I'm going to show are cross-validated. Yeah?
But that always assumes that the target is moving at the same rate, right?
The target is fixed. So the target is shown at the beginning of the trial, and it's fixed.
But you have to aim for a moving target.
It's fixed. Only the cursor that the animal is controlling is moving.
Okay.
Yeah.
Okay.
Yeah. Okay. So here is an example held out trial. Okay, so here the target is here, and the cursor, okay, was moving along this trajectory. Okay, and so let's just look at this time point right here. Right, this is where the cursor is now. So remember that at this moment in time, the animal doesn't know that the cursor is actually here. Okay, and the neural command that the animal sends at this time point sends the cursor in this downwards direction, okay? And that's a 45-degree error because it misses the target by 45 degrees, okay? In comparison, right, if we take the internal model that we estimated, right, and we say, okay, what would the animal do at this time point? Well, it'd go back 3 time steps, right, to its most recent sensory feedback, and then propagate that position forward in time by 3 time steps. It comes up with an internal estimate of where the cursor currently actually is, right, which disagrees with where it is on the screen. Okay? And so in this particular example, right, the neural activity that the animal sends, right, goes straight to the target according to where the monkey thinks the cursor is. Okay, so this would incur 0 degrees error. Okay, so now we can compare these 2 different types of errors, right? The error that actually occurred on the screen, which was 45 degrees at this time step, versus the error that was in the monkey's mind in some sense, right? Which in this case was 0 degrees. Yeah?
How do you estimate what is in the monkey's mind? So
Yeah.
you basically assume at every stage monkey thinks the cursor is pointing directly to the target.
In the training trials, we needed to assume that, right, to get the parameters of the internal model. But then this is a held-out trial, right, which was not used in model fitting. And so what we did was to take those parameters and the neural activity that we recorded on this trial to then propagate this sensory feedback forward in time to arrive at this point. Right? And then we use the neural activity that we recorded at the next time point to then ask where does that send the monkey's internal estimate. Okay. All right. So we can look at these whiskers, right, across all the time points, right? And the thing that jumps out at us, right, is that most, if not all of these whiskers point straight to the target, right, even during times when the cursor is not going straight to the target on the screen, right? And so before we did this study, right, we would sometimes see trials like this, right, where the cursor goes off in different directions that don't seem to make sense in terms of the task. Okay? But having done this, having now interpreted, right, these same trials in terms of an internal model, it all seems to make sense now, right? In that the errors that the animal was making was not due to some sort of random noise, right, or some moments of lapses of attention, right? What seems to be going on is that these errors are structured, okay, and that it's because the animal was using an internal model that was mismatched from the actual BCI mapping. Okay. Right? So under this interpretation, right, it seems like the animal is acting rationally, right? Even though without seeing these red whiskers, we would have thought, just looking at the black trace, that the animal were somehow acting irrationally. Okay, so that was one held out trial. We can look at a bunch of held out trials, right? And the thing that really stands out here is that there are times when the cursor is going straight to the target, and during those times, the whiskers align with where the cursor is actually going on the screen. And then there are times when the cursor is not going straight to the target, and during those times, the whiskers point to the target, right, and don't align with the actual cursor trajectory. Okay? And so it seems like across all of these trials, right, whether or not the cursor is actually going to the target, right, what the animal internally thinks is going on, right, is that it's always trying to push the cursor straight to the target. Okay, so we can quantify this, right? So we can measure these angular errors, okay? And what you see is it's a vertical axis here is angular error, right? The first bar here is the cursor error, which is the error of what the animal actually saw on the screen. And then the second bar here is the error according to the internal model. Okay, and what we see is that by interpreting, right, the population activity in terms of the internal model, we can account for over 65% of the errors that the animal is making on the screen. Okay, so the key idea, right, this is the key result, right, and so the main message here, right, is that when there's a mismatch between the animal's internal model and what is actually going on in the outside world, right, you can get errors from that. And in this particular task, we can account for a large percentage of the errors, right, by accounting for this mismatch. Okay, so I don't have time to get into additional analyses that we did, but let me just tell you what they are. So we also found that when we changed the BCI mapping, right, that the internal model also adapted in accordance to the new mapping. Okay? Secondly, we found that subjects attempted to move more slowly during hold periods and more quickly during movement periods. Okay? As if this mismatch between the internal model and the BCI mapping, right, actually decreased the dynamic range of speeds. Right? The animal had some trouble stopping and some trouble moving quickly, right? And we could see that by interpreting the neural activity in terms of an internal model. And furthermore, this mapping, right, from the high-dimensional neural activity to low-dimensional cursor kinematics was essential to the explanatory power of this internal model, right? And so we can see that in this equation, right, that defines our internal model, where the key term here is this b tilde ut. Right? So b tilde here is short and fat, right? And u is a vector of the neural activity, right? And so this operation of B times U, right, is a mapping from this high-dimensional neural space down to a low-dimensional kinematic space. Okay? And this mapping was critical, right? And we assessed this by replacing this neural activity with cursor velocities, right, that were actually shown on the screen. And we found that we could not explain the errors even close to the extent that we were able to by using the neural activity. Okay, so to summarize, right, what I've shown is that BCI facilitates the study of internal models because we can view it as a simplified closed-loop motor control system. We developed a statistical framework for estimating a rich internal model, right, from neural population activity, right, that operates at the time scale of tens of milliseconds, at the spatial scale of individual neurons. And we found that a mismatch between the internal model and the actual BCI mapping can account for 65% of the animal's errors, right? And then finally, the sort of broader message, right, is that population activity can provide insights about the internal model that may be difficult to see through behavior alone, right? It's because the neural population activity reflects these multidimensional thought processes. All right. That's it.